Development of zygote in flowering plants, germination of seeds, adaptive features in a developing animal. At the end of the lesson, you should be able to know the following. 1. Fertilization and development of zygote in plants. 2. Formation of seeds and fruits. 3. Germination of seeds and condition necessary for germination. 4. Adaptive features in a developing animal. 5. Definition of oviparity, viviparity and ovoviviparity. Development of zygote in plants. In flowering plants, the process of pollination is followed by fertilization, leading to the development of the zygote. This development eventually transforms the flower into a fruit that encloses the seeds. The male and female sex cells responsible for zygote formation are the pollen grain and the ovule, respectively. The following steps outline the processes involved. 1. Pollination. After pollination, the pollen grain lands on the stigma and absorbs a sugary liquid, causing it to swell and germinate. 2. Pollen tube growth. The outer coat of the pollen grain splits, and a pollen tube grows out and down inside the style. 3. Division of nucleus. The nucleus of the pollen grain divides into two parts: a large tube nucleus and a smaller generative nucleus. The male nucleus acts as the male gamete. 4. First fertilization. One of the male nuclei fuses with the ovule after its release into the embryo sac, resulting in the formation of a zygote. This zygote develops into the embryo, marking the first fertilization. 5. Second fertilization. The second male nucleus fuses with the secondary nucleus, forming the endosperm nucleus. This process leads to the production of endosperm, which serves as the food storage for the embryo. Formation of seeds and fruit. Development of embryo. The zygote undergoes division through mitosis, forming multiple cells that differentiate and organize into an embryo. The embryo consists of the following parts: A. Plumule, the embryonic shoot; B. Radicle, the embryonic root; C. Cotyledon, one or two seed leaves; D. Endosperm. This may or may not be present and serves as a food storehouse for the embryo. As the embryo develops, the ovary and ovules undergo changes and develop into fruits and seeds, respectively. Seed. A seed is the ripened, fertilized, and developed ovule. It comprises the following parts: A. Seed coat, the outer protective membrane of the seed; B. Hilum, the point of attachment of the seed to the seed stalk; C. Micropyle, the tiny hole through which air and water enter the embryo of the seed; D. Embryo, the innermost part of the seed. Seeds with a single seed leaf are known as monocotyledons, such as maize, millet, rice, sorghum, and wheat. Seeds with two seed leaves are called dicotyledons, such as mango and beans. Germination of seeds. Germination is the gradual development of the seed embryo into a seedling or young plant. Embryos of developed seeds often undergo a period of rest called dormancy, until favorable conditions for germination arise. There are two types of germination. 1. Epigeal germination. The seedling emerges from the soil with the cotyledons above the soil surface. This type is observed in dicotyledonous plants. 2. Hypogeal germination. The seedling emerges from the soil, while the cotyledons remain below the soil surface. This type is observed in monocotyledonous plants. Conditions necessary for seed germination. For seeds to germinate successfully, the following conditions are required. 1. Adequate supply of water or moisture, which softens the seed coat and activates the soil for further development. 2. Oxygen for respiration and energy generation necessary for seed growth. 3. Optimum temperature suitable for seed germination. 4. Enzymes to accelerate the breakdown of food and release energy. 5. Food or energy stored in the cotyledons for dicot seeds or endosperm for monocot plants. 6. Viable seeds capable of germination. Adaptive features in developing animals. Eggs of fish, amphibians, reptiles, and birds. Vertebrates other than mammals reproduce by laying eggs. The eggs possess several features that facilitate embryo development. 1. Shell. A hard, porous structure rich in calcium carbonate that provides protection and aids in respiration. 2. Membranes. Additional protective layers surrounding the egg. 3. Albumin. Egg white, which accounts for over 50% of the egg and provides nourishment to the developing embryo. 4. Yolk. Located at the center of the egg, it is rich in vitamins, protein, and minerals, serving as a nutrient source for the embryos. 5. Chalaza. Thick protoplasm extending from both sides of the yolk, anchoring the embryo in position. Developing embryo fetus in mammals. Embryos or fetuses in mammals possess the following adaptive features. A. Uterus. After fertilization in the oviduct, the embryo implants itself in the uterine wall. B. Placenta. It forms the connection between the embryo and the mother in mammals. The placenta transports oxygen, water, and nutrients from the mother's blood to the embryo and eliminates waste products from the embryo's blood. C. Umbilical cord. It attaches the embryo to the placenta, carrying the blood of the fetus to and from the placenta. The mother's and fetus's blood supplies remain separate. D. Embryonic membranes. These membranes envelop the fetus, providing protection. They include the amnion, filled with amniotic fluid for cushioning, chorion, absorbs oxygen for the embryo, and allantois, facilitates respiration and excretion. Definition of oviparity, viviparity, and ovoviviparity. 1. Oviparity. Reproduction where eggs are laid in the external environment, allowing the embryos to complete their development and hatch from the eggs. Most fishes, amphibians, reptiles, and some birds undergo oviparity. 2. Viviparity. Reproduction in which the young develop and are nourished inside the female's body through the placenta, and they are born alive when fully developed. Some fishes, reptiles, and all mammals exhibit viviparity. 3. Ovoviviparity. Reproduction occurs in certain fishes and reptiles, where the laid eggs are retained in the female's body without a placenta. The young hatch from the eggs and are subsequently released. Summary. In summary, we have to end this lesson with a few summaries of today's lesson. The main highlights are: 1. The process of zygote formation in flowering plants involves pollination, fertilization, and the development of the embryo. 2. A seed is a ripened, fertilized, and developed ovule. 3. Refer to the labeled diagram for a detailed description of a seed. 4. Germination refers to the gradual development of the seed embryo into a seedling or young plant. 5. Epigeal germination occurs when the seedling emerges from the soil with the cotyledons above the soil surface, while hypogeal germination involves the seedling emerging with the cotyledons below the soil surface. 6. The conditions necessary for seed germination include adequate water supply, oxygen, optimum temperature, enzymes, food or energy, and viable seeds. 7. The labeled diagram describes the adaptive features of the egg in birds, including the shell, membranes, albumin, yolk, and chalaza. 8. Oviparity involves laying eggs externally, viviparity involves live birth after internal development, and ovoviviparity retains eggs internally, but lacks a placenta. Examples of oviparous, viviparous, and ovoviviparous animals include reptiles, mammals, and certain fishes reptiles, respectively.